Welcome everybody. It's noon here in the East Coast and I am delighted to welcome you. I'm Carla Rothling and I'm here with one of the co-organizers of the Global IMMUNOTALKs, Enrique Veiga Fernandez, who will be the one introducing our speaker today, Gabriel Victora. So welcome Enrique, welcome Gabriel. Enrique.
Thank you, Carla.
The microphone is yours.
Thank you, Carla. So it's really a pleasure to welcome you to this talk, part of the Global IMMUNOTALKs of 2021. It is my tremendous pleasure to introduce the speaker of today, Gabriel Victora, a friend of mine for quite a long time now. He's currently assistant professor at the Rockefeller University and also head of the Laboratory of Lymphocyte Dynamics in the same university. And today we'll give a talk on clonal dynamics of B-cell response. So Gabriel, he was actually born in Porto Alegre in Brazil. So we share something in common with Gabriel, the same mother tongue, Portuguese. One very interesting aspect that perhaps some of you are not acquainted with Gabriel past is that he actually initially trained as a musician and actually even became a very accomplished, I have to say, pianist. So if you don't trust me, just go to YouTube and check Gabriel Victora and you will confirm what I'm just saying. But then later on, actually, he started his studies more deep into biology, in particular in immunology. And so he obtained a PhD in immunology, where he studied B-cell biology in the New York University and Rockefeller University, where he trained with Michael Dustin and Michel Nozesweig. And there, I mean, he made very, very important contributions to the understanding of B-cell biology. And already from that very early days, he actually showed a raw and rare talent to develop new technologies. So he developed new techniques using intravital microscopy, genetic models and photoactivation that really completely transformed the way we looked at B-cell biology in general and specifically in germinal centers. Then he became, as an independent investigator, a Whitehead Fellow at the Whitehead Institute, MIT, in the US. And he really there started really pioneer and continued pioneer work on B-cell clonal dynamics. I'm sure he will share some of those exciting recent data with us today. But not only on B-cell dynamics, he also started to explore new territories, and notably on interactions of B cells with T cells, and how these cellular and molecular interactions could contribute to B-cell clonal selection at a cellular and molecular level, obviously. And then more recently, he started also to expand these studies and cell-cell interaction, understanding how T cells interact with dendritic cells, and you likely are all aware of this wonderful paper they published a few years back about the lipstick technology and how actually to track and understand intercellular interactions and relationships. So it comes to no surprise that his work has been published in top journals, including Science, Cell, Nature. And certainly for those reasons as well, we received multiple accolades and awards. He's, for instance, a Searle Scholar. He received recently Boros Wellcome Fund Investigator, a Pew Stewart Award, he's MacArthur Fellow, and an NIH Director's Pioneer Award. So Gabriel, it's really a pleasure to welcome you today with us, and welcome to this Global IMMUNOTALK 2021.
Thanks very much for the very generous introduction. It's really a pleasure to be here. And congratulations for this very important initiative, something to keep us abreast of what's happening with, during this pandemic.
Wonderful. So one of the already sort of classic aspects of these talks is to actually ask you a question a little bit outside the talk. And actually, I would like to ask you something that I believe is quite important and perhaps inspirational, particularly for the young audience that is attending this seminar. If you would have to select one specific unknown that is exciting the most at the moment in cell biology, what would that be? And secondly, what are the technology hurdles that have not allowed you to tackle that specific unknown at the moment?
Well, I think to me the most interesting, biggest problem that is around that I really don't know how to answer, though we are trying, I think, as you'll see when we talk, to somehow get to this, is how is it that B cells choose what epitopes they like and don't like? What does immunodominance mean? What makes an epitope something attractive to a B cell and others not? I think treating B cells as individual clones with different specificities is a very important step toward this. This is what we try to do. I kind of grew up in sort of thinking of B cells like most B cell biologists do, sort of as a population, and looking at the genes that are important for this or that, the interactions, the cytokines, the T cell help and things like that, but as B cells as a cohort. And that kind of, in a way, ignores the fundamental problem of which clones do better than other clones, which I think, for example, the HIV crowd and the influenza crowd, by sequencing human antibodies and showing us where they bind, have really brought to our attention that this is an important part of B cell biology. I think the technology that is really missing for us to be able to tackle this properly is a good high throughput way to match the clonal sequence with the specificity. We can sequence thousands and thousands of cells, but we can't make thousands and thousands and thousands of antibodies. And I think people have been working on this, and I think you know, there are different technologies, antibody production in larger scale, and yeast display, and single B cell cultures, and I think we're getting there. But I think this will be transformative when we learn how to take all our sequence data and quickly assign it to epitope specificity.
Wonderful. Thank you so much, Gabriel. So it's a real pleasure to give you the stage, Gabriel, so you can share your talk with us. And so, as I referred at the very beginning, Gabriel will be showing data from his lab related to clonal dynamics of antibody responses. Thank you, Gabriel.
Okay, I'm sharing the wrong screen, right? This looks right?
Maybe.
Yes.
Can you go? I think so. Yeah. Can
I think it does look right. Yes.
You see one slide or two?
One.
One. Okay, good. So that's right. Okay, thank you. So yeah, I touched a little bit on what I want to talk about, and what I think is probably, to me, is the most interesting question in B cells, which is how B cells behave when you think of them as clones. And this is what we call clonal dynamics. And I'll spend a few slides in the beginning, try to phrase how we have been looking at B cells for a long time, and then sort of introducing how we want to look at them and what the tools are we developed to do this before I get into more recent work that we've done on this. So I'll start by saying that antibodies are not a new problem. I don't think this is a surprise to anyone. We've been as a field looking at antibodies and thinking about them for over 100 years. But I think a lot of the work has been focused on what I'm going to call here the original antibody problem. To illustrate this, I like to use this drawing by Linus Pauling, not only because it shows you how old the problem is. This drawing was made in 1940, and this was already 50 years after we started thinking about these problems as a field. But also because it illustrates very well the things that have to be reconciled in order to understand antibodies. And I think three main things are relevant here. One is the very best antibodies, the ones we really want to generate in vaccines and to use as therapies, are very high affinity. They bind very strongly to their antigens. And this is illustrated here by this very snug sort of lock and key fit between the antibody here on top and this hapten on a protein here at the bottom. So in order for antibodies to be good, they have to bind things very, very strongly so they don't come off. The other thing that antibodies have is narrow specificity. And this is illustrated here by these opposite charges. If you have a negative charge here on this tip of the hapten, you need a positive charge on the antibody. And if it were reversed, if you had a positive charge on the hapten, you'd need a different antibody, not this one. This one wouldn't bind. So antibodies, when they're very good, they tend to have sort of narrow specificity. At least they are able to distinguish between fairly closely related antigens. A corollary of this is that if you have unlimited antigens, you need unlimited antibodies, which means there are many, many different antibodies that you need to be able to make if you're going to bind anything, even these invented hapten molecules here that don't exist in nature. So these three things together can be summed up in sort of a 21st century question, which is how are you able to code for an unlimited diversity of antibodies using a limited genome? Right. Now, we know a lot about this. The first part everybody, I think here knows, antibodies are not single genes. They are assembled from many different segments during B cell development in a clonal fashion. So by assembling hundreds of V's and some other segments during B cell development, which make up the variable portion, the antigen binding portion of the antibody, you end up with millions of combinations that then can be selected for by antigen, and you'll usually find some combination that is good enough to start a reaction. What is not always as appreciated is that these original VDJ recombinants are not usually very good at binding antigen. For them to be good, they have to go through this second process here, which was described by Hermann Eisen in the 1960s, which is affinity maturation. And what Hermann showed was that by measuring the mean affinity of the antibody in the serum, these are not titers, these are how good the antibodies actually are per mole. You see that antibodies don't start out as good. If you look at 2 weeks after an immunization, antibodies are sort of in the micromolar range, this is not great. But if you just wait, you don't have to re-immunize a rabbit in this case to see something like 100-fold increase in global affinity in the serum at 8 weeks. And this is sort of the second phase. On top of the VDJ rearrangement, we have this learned part where the antibodies learn how to recognize antibodies better and better. Now, since Hermann described this in the 60s, we have made a lot of progress in understanding how it is that antibodies do this. I'll give you a quick overview here. What you start with is a pre-immune repertoire that is the product of VDJ recombination, many different clones, millions of different clones, or even more. When antigen comes in to the body, it selects from this initial compartment, not something that binds to it with high affinity usually, but the best clones that it can find around. So I'm coding this in by color here. If you're a red antigen, you come in here, you select this sort of close to red B cell clone because it's the one that binds the best to the antigen. And even though it's not perfect. But what this does is it triggers this B cell to expand and become a clone. Part of this clone differentiates into plasma cells, and this secretes that early wave of antibody that comes up at 2 weeks after immunization in Hermann Eisen's graph. But another part of this clone goes back into the center of the B cell follicle, starts to proliferate very rapidly. And then it does something really amazing, which is this clone starts to mutate itself. It expresses this enzyme activation-induced deaminase that introduces mutations into the VDJ region, into the antibody binding part, antigen binding part of the antibody. And these are random mutations, so that a lot of them are deleterious to the B cells, but occasionally you find a mutation that improves affinity. Now, inside the germinal centers, there is antigen retained as immune complexes, and this antigen serves as a selection platform, so that the best random mutants can be selected and expanded in the population. And this is a very Darwinian sort of framework. It's actually has the 2 tenets of Darwinian selection, which is a descent with random modification followed by some form of fitness-based selection. If you do this over and over, you increase fitness in the population. And as these cells become fitter, they export plasma cells that now go on to secrete the high affinity antibody. That is what you see at the 8-week time point in Hermann's graph. So this has been very much framed as a Darwinian event, and I agree with this framework. Now, what this graph figure does not contemplate is that antibodies are actually very diverse. This is sort of looking at a very monomorphic kind of phenomenon where one clone comes in, the high affinity variants of it are selected, and this leads to high affinity antibody. But it ignores what I'm going to call now the other antibody problem, which is how do antibodies choose what they're going to target? And you can use haptens as a good example of this. If you take a hapten like this and you attach it to a protein, which is what you need to do to get a good immune response from it, even if you attach only very few haptens to this protein, the immune system will normally prefer to target the hapten, the small hapten, over targeting this huge open area of the protein. So haptens are known to be immunodominant. Why it is that the immune system prefers the hapten over the large surface of the protein is, I think, one of the more interesting problems that are out there for B cells. And it's not only interesting biologically, it's actually very important for health, because if you think, for example, of what we want to do with B cells, which is make a good broadly neutralizing vaccines to influenza and HIV and also maybe coronaviruses in the future. One thing that is seems to be, let's say, a common feature of many of the antibodies that are broad neutralizers is that they tend to target non-immunodominant epitopes like the stem of influenza and the CD4 binding site of the HIV GP120. And even though they do that, they are actually very high affinity antibodies that are very highly mutated. So the question of how do you take a B cell that responds to a non-immunodominant antibody and keep it within germinal centers, maturing for long enough for it to become a very good B cell, is something that's not included in that model that I just showed you. It's not contemplated by that framework. Now, so this sort of problem of interclonal competition, clones competing between and with each other in a mixed germinal center, is I think a whole order of magnitude harder than the problem of intraclonal competition, where just affinity is the main driver. And I sort of like to use a kind of a silly evolutionary analysis, since we are on the Darwinian theme, to just give you a sense of how much harder the problem is. That what we have been doing so far is a bit like evolutionary genetics. It's sort of similar to the giraffe neck problem, the classic giraffe neck problem. We know what the incentive is. The incentive is to be able to eat leaves high up in trees. If you have two giraffes and one has a longer neck, this one will eat the leaves, this one won't. And the giraffe with the shorter neck won't leave as much progeny as the one with the longer neck. And this is how you increase fitness in a population. Very simple. Now, what we're trying to do looks more like this. If you think that leaves on a high tree are not the only thing that animals are competing for, you go into sort of a quantitative ecology problem. You think, well, leaves might be a limiting factor at some point, but at some other point, maybe water is a limiting factor, and then the giraffe is going to have more trouble getting to this water. And this really is a very difficult problem to deal with. And a lot of what we have been doing in the past years is trying to make this into a tractable problem that we can somehow gain insight into, even though it is very complex. So I'll talk a little bit about how we started on this for two reasons, just to introduce a couple of concepts and also a couple of tools that I'm going to refer to repeatedly. But this started out when I was at the Whitehead Institute. Two people were very important here, Jeroen Taas, who was a Dutch master student who came in to study this in the lab and is now a PhD student at Harvard, and Luca Messen, who was a PhD student in Norway in Ludwig Solid's lab. He came to the lab and now he's a research associate. He's sort of the leader of the sequencing and clonal effort in our lab these days. What we did when we came together was try to start to understand what stage were we at in understanding the clonal dynamics of the antibody response. And this is sort of what we could come up with at the time. This is a review we wrote in 2014, where we started drawing these little colored circles that we do a lot of now. And what was known at the time was that, well, there are lots of cells in the pre-immune repertoire that are capable of entering an immune response. So if you take some cells, they might be very low affinity, you put them in a mouse where there's no competition, these cells will respond and make beautiful germinal centers. So there are many cells that can respond, but these cells are limited by bottlenecks. So the first bottleneck, the first fact that or proposed idea that was out there was that germinal centers were polyclonal. And that they began with something like 3 clones and 6 clones or something like that. And this implied that this bottleneck here was very, very strong, that you'd go from thousands of clones to a very small number of clones. And then a germinal center selection with AID, et cetera, would come in, and this could lead you to a further reduction of clonality in late GCs. Now, we were interested in understanding if this was really the case, because it seemed to us very difficult to have such a strong bottleneck where the GC fate would be restricted to only the very few best clones, which sort of seemed to defeat the point of having a GC to begin with. So understanding whether this was really the case required us, oh, sorry, I always forget this little star here. It's going to show up a few times. This was bad news for our flu stem binder, for example, because if it's low affinity, it's never going to be able to get into a GC to begin with. Right? So in order to understand this bottleneck number 1 here, I think one thing that we thought was very important to be able to do was count how many cells, how many clones are in an early GC very precisely, instead of inferring it from indirect methods as was done before. This, Enrique referred to you, a good way to do this is to use photoactivation. And the rationale here is that if you take a lymph node, there are many germinal centers in it. If you want to count how many clones are in a single germinal center, you have to be able to separate it from all the other germinal centers in the lymph node. And this is a recurring theme in our work. One way to do this is to use in situ photoactivation. You take a photoactivatable GFP transgenic mouse that we described in this paper here when I was a PhD student. And you take a lymph node from this mouse that has germinal centers, you use your laser to paint this germinal center green. And then when you go to the flow cytometer, if you take green cells out, you're taking cells from one germinal center and not from all the others. And then these cells can be taken out and put in a 96-well plate. And using Michel Nussensweig's methods, we can sequence the V gene sequence and determine the clonal identity of this cell. Now, what we found when we applied this to very early germinal centers is that they are actually quite diverse. If you look here, this is 6 days after chicken gamma globulin, one of our favorite model antigens, in alum immunized in the footpad. If you take 2 germinal centers from popliteal lymph nodes, you'll see that, for example, here, these pie charts we use a lot. Each of the slices in the pie chart is a different clone. They're colored randomly here. Out of 77 cells that we sequenced from this germinal center, we found 46 different clones. And this we found over and over. So it seemed like the early germinal centers were actually quite diverse. And if we looked here on average, you can use equations from ecology, from quantitative ecology, to try to understand what this means, what this sample means for the whole germinal center. And if you extrapolate this to the whole germinal center, these numbers tell us that germinal centers are founded by something in the neighborhood of 100 clones, even when you immunize with a simple antigen like a chicken gamma globulin. So what this is telling us is that bottleneck number 1 is not that strong. It allows quite a lot of clones into the germinal center. And this was very important to us because if you're thinking of the germinal center as a ecological competition between many different clones with different incentives, the first thing you need is for the germinal center to be seeded by many clones for this to be an interesting question. Now, okay, germinal centers start out very diverse. How do they lose this diversity over time? And to look at this, we started using a different technique, which was made possible by the neuro folks at Harvard, Jeff Lichtman's lab, who came up with this really cool rainbow technology, where you use Cree recombination to make cells express one of a number of different fluorescent proteins, sort of stochastically. And once they fix their selection of what protein they're going to express, they transmit this to their descendants. So it's sort of a permanent labeling in many different colors. This is the allele we use from Hans Klever's lab. It's called confetti. It is in the ROSA 26, so it's expressed in all cells. And when cells express Cree in some form and excise the stop codon, they can pick between these 4 fluorescent colors. And if you have 2 alleles of this, you end up with 10 different color combinations that you can then trace over time as clones expand and contract in germinal centers. So if you use this with AID-CRE ERT2, this mouse was made by Jean-Claude Weil and Claude-Agnès Reynaud in Paris. You can fate map in these colors exclusively in the germinal center. So if you start germinal centers prior to tamoxifen administration, they're all black. But when you use tamoxifen, administer tamoxifen to these mice, they end up, cells end up picking colors randomly. And if there is selection, you'll see that very clearly as cells of one color taking over the whole germinal center structure as the germinal center loses its initial diversity. So this works very, very well for germinal centers. This is just an example. If we do this, this is 5 days after initiating germinal centers. We gave tamoxifen to a mouse and we're looking 3 days later. This is an NP-CGG immunization again. So 3 days after tamoxifen, there hasn't been much time for selection to happen. And you can see that here. There's 5 germinal centers here in this popliteal lymph node, and all 5 of them have all 5 colors. So they are very mixed, and this is expected because they haven't had time to select. Now, if instead of looking 3 days after tamoxifen, you look 15 days after tamoxifen, you start to see that selection has been happening. And this is very clear from these 2 germinal centers here that went from a mixed phenotype to a very much single colored phenotype. But at the same time, we see that this is not the norm. But most of the germinal centers tend to retain quite a bit of clonal diversity. And in our sort of analyses so far, when you do this with immunization, it seems that something like 10% of germinal centers are able to do this and select heavily into one dominating clone, while others maintain their diversity. Now, to understand how this happened at the sequence level, it again would require us to isolate this particular germinal center here, as opposed to all the others. We couldn't use photoactivation here because green is one of the colors in rainbow. But what Jeroen figured out how to do was to slice these germinal centers on a vibrotome, slice the lymph nodes on the vibrotome, which allows us to keep the cells alive. And you take these slices, you put them in the scope, you find a corner of a slice that has only one germinal center, and you dissect it out with a scalpel on a dissecting scope. And what you can do then is put this little slice into a tube and sort cells from it. So you can sort cells from this little bit of a slice that allows you to obtain sequences from the germinal center of interest. And when we did that, we started to see what it took to become single colored. And what really we think happens in these cases is what we started calling a clonal burst. So this is what we were able to sequence from day 10 post-tamoxifen germinal center. This was a very, very single colored CFP GC here. Over 90% of cells belong to this clone. And what you see here is if you take the cells that were here and you make a clonal phylogenetic tree from the sequences that we get them using the somatic mutations to guide you, what we see is in this case, and also in most cases when we have single colored GCs, what happens is this thing we call a clonal burst. And you can see this here. You start from the unmutated ancestor, you made 1, 2, 3 mutations in the heavy chain, and you hit sort of a jackpot. And this we can see this because this number 17 here means that we found this particular sequence in 17 different cells in this germinal center. And everything else seems to be a descendant from this cell. So this basically tells us that one single cell got a really, really strong signal, went to the dark zone, divided something like 10 or 12 times in a row until it took over the whole thing. And this is how we think that most diversity is lost in the germinal centers from these clonal bursts. And these are usually associated with higher affinity. So in this case here, this clonal burst, we measured that it was something like 15-fold better than what the cell started with. So we've revised the model a little bit. We start with the pre-immune repertoire that has many responsive cells. We know experimentally that this bottleneck exists. We just don't think it is as strong as was previously proposed. So that early germinal centers start with something like tens to hundreds of different clones, which leads us to an interesting sort of competition between clones that I think was not completely appreciated before. This can be resolved in different ways. Germinal centers can basically resolve very slowly in the sort of low burn mode, where there's slow selection, not that much affinity maturation, but occasionally you get these clonal bursts where all the diversity is wiped out and you remain with one single high affinity clone. And of course you have all of, this is a gamma of different phenotypes. It's not two different types of germinal centers. You have the whole range of phenotypes from the low grade selection up to the clonal burst. And this in our experience happens in about 10% of cases. And this was sort of good news for our starred stem specific clone here, because we know that it doesn't get blocked out from entering germinal centers. It can probably make it in and evolve in these slow evolving germinal centers and accumulate mutations, which is a good starting point. Now, of course, germinal center cells are not the end of things, right? The germinal center cells don't secrete antibody. In order to be able to secrete antibody, you have to be exported from the germinal center, either as a plasma cell or as a memory cell that then gets activated later to become a plasma cell when you see the antigen again. And we started to think more about these post-germinal center fates and what would happen to our stem-specific clone in these cases. I'll tell you a little bit about this fate here, memory, quickly, because this paper was published about a year ago. Again, this was the work of Luca Messin in the lab, this time helped by Arjen Skeepers, a PhD student from the Rockefeller program. And what Luca and Arjen set to try to understand is what is the relationship between the primary response and the secondary. This is here Immunology 101. We know that when you start to give a prime, you get a little bit of antibody, but the real strong antibody response you get happens the second time you see the antigen, when you get a much higher peak, which is sort of logs higher than the first one. And I'm sure you've seen a lot of this lately in graphs from COVID vaccine trials, that the second exposure is really very important for protection. What we wanted to ask was, what was the clonal dynamics of the secondary response? And thinking of it back in our sort of little colored circles diagrams, if you go from a high diversity, low affinity germinal center towards a low diversity, high affinity response with where these clonal bursts are more evident, you're going to export memory throughout this. And we know from the work of Mark Schlomchik that most memory comes out early in the response. And this creates sort of a lopsided diversity. So you have a lot of the diversity coming up later, but the better cell earlier, but the better cells come back later. And what he asked was simple: what do you get when you boost? Do you favor the low affinity but very abundant early memory, or do you favor the late higher affinity memory? And how does this work out in the plasma cell population that you get? The other question, of course, was we know from work, again, of Mark Schlomchik, also Mark Jenkins and Claude Agnès and Jean-Claude Weil, that germinal center B cells, after they exit a germinal center's memory, they can be recalled to germinal centers again. And this is very clear in sort of transfer models that the cells have the potential to do this. We wanted to try to understand what is the clonal dynamics of this also in a single mouse without a transfer. So I'll start by telling you a little bit about this. We adapted our tamoxifen model that I just showed you about, that I just showed you the data for, to a prime boost model. What we did was, here with chicken gamma globulin, we primed one foot pad of the mouse. We then gave tamoxifen to fate map the germinal centers in this response. And then a month later, we boosted the other foot of the mouse. And the reason we did this here has nothing to do with should you boost your left arm or right arm with the coronavirus vaccine. This was actually, we wanted to be sure that when we boost, we start with a naive lymph node without ongoing germinal centers. And we know from our experience that germinal centers last for a very long time here after this chicken gamma globulin injection. Certainly at 30 days, they're still there. So we didn't want to reboost germinal centers. We wanted to start again from scratch. And then we imaged this left popliteal lymph node to look for cells that had come from the right foot pad. And what we saw kind of surprised us at the time. Now we're getting more used to it. But what we did see was that very, very little of the secondary response came from cells that were in primary germinal centers. So there's several germinal centers here. We don't see them because I'm not showing you the follicular dendritic cell stain, but there are several germinal centers in this mouse. One of them we blew up here, and most of them look like this, not a single colored cell to be seen inside the germinal center. Now, this is not to say that memory cells do not re-enter germinal centers. They clearly do. They're here. So there are 3 germinal centers here that have cells in them. This is just a very inefficient process. So to tell you, give you an example, a quantitative example of this, when we did the primary fate mapping, we fate mapped about 70% of cells, but when we recalled these cells, we got something like 4% of secondary germinal center cells came from a primary germinal center. Now, another thing that was interesting here is very striking in this particular image is that different colors go into different germinal centers. So there's a blue one here, a red one here, and this one has green and purple. And what this tells us is that it's not only few cells, it's very few clones. If to get the pattern like this, you have to be starting from a very, very limited clonal diversity in the secondary germinal center. And we can quantify this. These are pie charts of clones with their colors assigned for whole popliteal lymph nodes, asking you which cells returned into germinal centers. And this one is the one I just showed you. You see a big blue clone, a big purple clone, a smaller green clone, and a small red clone here, just as the picture suggests. What you see from this is that very few clones dominate the entire response. Something like 5 or 6 clones on average constitute the bulk of what comes in from the primary germinal centers to the second. So there's a big bottleneck here. And this is not to say that these cells don't contribute to germinal centers. This one here, for example, if you sequence the B cells, you see that this memory cell here was recalled into the germinal center, and it's obviously expanding here. There are 7 of them that we sequence and further mutating and leaving descendants. So it's clear that memory cells do re-enter. They just, in this one-shot prime, one-shot boost model, they seem to constitute very little of what you are getting back into the GC. This is also not to say that there was no memory there. If you actually look for the boost effect, you would think, well, this must come from memory plasma cells, and this seems to be the case. If you look at the medulla of this lymph node here, you see that it's packed with colored cells, and this is where plasma cells that were induced by the boost tend to live. And you can quantitate this as well in this model. This is actually using Tomo Kurosaki's S1PR2 Cre-ER tomato. It does the same thing, but it's more efficient. If you fate map germinal centers using this mouse, you get almost 100% of cells fate mapped. But when we boost, we're down again to 5% in the boost germinal centers. But in the plasma cell compartment in this regimen, we kind of match the labeling efficiency. So there's a lot of plasma cell responses, and most of the plasma cells come from memory B cells from the primary, as expected. What is interesting, though, is that the bottleneck still applies to these plasma cells. So I told you that 6 clones constituted the bulk of what we got back into secondary GCs. If you look at what's in here in these plasma cells, it's actually even lower. About 3 clones constitute the bulk of what you recall into plasma cells. So really, really strong bottleneck. And this is sort of the major conclusion I wanted to derive from this here. Is this bottleneck idea. So in secondary germinal centers, these are mostly devoted to primary diversification of a new, which we think is likely naive repertoire. GC experienced memory B cells can re-enter, and they will do this probably according to their numbers. So if they're more memory, they're going to be more memory. But in this single prime boost model, they do so inefficiently. The recall response is very restricted. There's this post-boost bottleneck, which leads to this question here that I'm going to go quickly through. Where is this missing clonal diversity? If we came in with hundreds of clones and we got out very few, where is this diversity? Quickly, we wanted to look at this in a model where we could track memory cells, because this is where the diversity would be.
Let's see. Somehow Enrique, could you hear Gabriel well, or did you also hear a sound? Yeah, let's see.
Yeah, I was getting some sort of noise, actually, some sort of interference.
Yeah, let's see, Gabriel. Let's see if you can talk again. I just want to make sure that people can listen you well.
Okay.
Let's see.
How is it now?
I can hear you well now. Okay.
Now it's good.
Sorry for the interruption, but it's so beautiful that want to make sure that people can hear well. Okay, let's try again. Thank you.
Let me know again if this stops working. I'm going to go a little bit quickly through this because time is running out. But there is a model where it's very easy to find memory B cells, which is influenza, because we have very good tetramers to track memory and cells that we are really sure are memory bind this tetramer, and they are also fate mapped. So we can do this. In here, what we started by infecting mice with flu intranasally. We labeled primary germinal centers with tamoxifen. These are the very nice pictures we get after 45 days of selection happening in the system. And then we boost the foot pad at 45 days and ask what from the memory from the primary comes back into the foot. And this is just to show that we get about the same as we get with NP, shipping gamma-globin immunization. We get very good labeling of germinal centers, very little re-entry of primary germinal center B cells into secondary germinal centers, and good plasma cell recruitment from fate map cells, although a little bit lower than we would in simple immunization. But the nice thing here is that using tetramers, we can find sort of this hundreds of B cells per germinal center that per lymph node that come from the memory. So we use this to sequence everything we can in this mouse. We sequence the primary lymph node, we sequence plasmablasts in the bone marrow, plasma cells in the bone marrow, plasmablasts in the boost, germinal centers in the boost. And we put together these drawings here that Luca is now very good at making, that we call clonograms. And these are sort of those pie charts, but standing on end. So each slice is a clone here. And we color these according to where they're found. So if they're gray, they're found in only one compartment. If they're colored, it means they're shared with different compartments here. And I'm not going to go into this in detail because there's very rich information here. I'm just going to point to a couple of things. The plasma cells here, the plasmablasts that we recruit from memory, are again very, very restricted in terms of their clonality. So you see that they're highly dominated by very few clones. And this is even though we did mobilize a very high number of clones into the response, because if you look here, you'll see that memory is extremely diverse. That here in the spleen, for example, of 186 memory clones we could find, we found 111 different clones. So the missing diversity seems to be housed in the memory. There is a strong bottleneck here between selection, between making memory and selecting it to come back as a plasma cell. Now, you can ask what makes the blue cells here, the ones that were able to come back, so special compared to the other ones that didn't. And what we do here is we operationally divide these cells into two types, what we call used memory and unused memory. And then we go on to ask what's different about them. And two things strike us from this. One is that unused memory seems to be early memory. So if you look at how many mutations the unused memory B cells have, most of them have 0, and a few of them have some more mutations. So memory that's made early tends to be used less when you boost. And if you look at used memory, though, it seems to evolve, seems to come from the whole history of that clone in the germinal center. And you can find used memory cells that are much more mutated than the unused memory. So they're exported later in the germinal center. The other thing we can find is that used memory B cells, if you revert them to their unmutated ancestors, they tend to be higher affinity. And this is what we like to, we like to think that the best cells get recruited into germinal center, and these tend to be the ones that are then usable by memory at a boost. So to summarize the second part, focusing on a few clones and limited germinal center reentry are also true in influenza infection. That's not only in our prime boost with model antigens. The missing diversity between what you get into the primary and what you get into the secondary seems to be found in this untapped memory B cell population that comes out mostly early. The responding memory B cells are exported throughout the primary germinal center, and they tend to derive from cells that started out already better. So starting out well is an important thing to ensure that you'll participate in the response for a longer time. So I'm going to do a last one of these models here with the little dots. I've gone through this. Pre-immune repertoire gets restricted over time, especially when clonal bursts happen. We confirm, I think very nicely what Mark Slomchik had shown us, that memory tends to be exported mostly early, and a huge part of the diversity that goes into the germinal center ends up in this early memory compartment. What we added to this is a twist that says that if you boost homologously with the same protein, you end up recruiting mostly cells that come out later and that derive from these better clones. And this is here. Now, secondary germinal centers tend to be composed of these clones that look naive. We can't be completely sure that they're not pre-germinal center, early germinal center memory. We like to think they are naive, but we don't have definite evidence for this yet. But there are clearly memory B cells in secondary GCs. They can continue to evolve, but they also tend to come from very few clones. And we expect that this is probably to do with the inefficiency in generating memory compared to how many cells from the primary repertoire are able to come back when you do a secondary response. And this might be part of the reason why we do this. But this post-boost bottleneck, which is sort of what I want to really emphasize from these data, seems to be bad news for our stem-specific yellow clone here. And finding out how to break this bottleneck and get these cells to come back later could be an important tool to have if we want to immunize and get non-immunodominant clones to move forward. So I'm going to try to now in the last 10 minutes or so go quickly through a more recent story that we just published, which I think really makes the full use of this idea of the ecological competition in germinal centers, which is what the question we wanted to ask is what really happens in the gut? Can you have antigen-driven competition in the gut? And why is this so important, I think, or so interesting to us? And one reason is just that the gut seems to be doing lots of things at the same time. There are many antigens here from commensals. You're negotiating with food-derived antigens where you may or may not be able to induce high-affinity antibodies to food. This is something unsettled, I think, in the literature. Now and then, our guts are infected with pathogens, and these sort of provide, sort of give an additional challenge to the germinal centers here in Peyer's patches and lymph nodes that are already dealing with all this diversity. So we thought this is really an interesting place to look at these problems of ecological diversity and competition between clones with different incentives. Now, to answer this question, can antigen-driven selection takes place in the gut? The answer is very clearly yes for this kind of scenario. When you have a monomorphic antigen like a vaccine or an infection, work from several people has shown that you can get affinity maturation to happen. So we didn't go there. What we wanted to understand is this here. Can you get a steady state affinity maturation when all you have is your commensals and food? There were reasons to think it wouldn't happen. GCs in the gut in steady state seem to be open to non-specific B cells. And one of the most striking examples is the BCR-less LMP2A mice made by Stefano Kazola and Klaus Rajewski's lab that go into these germinal centers even though they don't have a B cell receptor. Albert Bendelak's lab and others have described that frequently you get polyreactive B cells, at least in the IgA compartment, as an output of this. Peyer's patches have generative roles. They are generating diversity prior to affinity maturation in other species. But in our view, the biggest challenge to making affinity maturation happen in these germinal centers was just the sheer diversity of antigens you have here. Germinal centers have to be able to negotiate between thousands of antigens and still pick a winner. And this was the question we had in mind when we started this. Another reason for me to do this was that when I moved to Rockefeller, I moved next door to my old-time friend, Daniel Musita, and we had been thinking about working on the gut on this germinal centers for a long time. So first thing we did when we got here was hire Carla Novosad, a postdoc in the lab, who did most of this work. She is now on the job market if anyone is recruiting. She was aided by our experts in sequencing and clonal dynamics in general, Luca Messin, who also trained actually in gut immunology in Ludwig Solid's lab in Norway. So we all teamed up and decided, let's try to tackle this complicated problem here. And we tackled a complicated problem with a very simple experiment. It's the same rainbow experiment we did, but even simpler because we don't immunize the mice. We take SPF AID Cree mice. They have germinal centers spontaneously in mesenterics and Peyer's patches. We give them tamoxifen, label cells in some colors, and just let them evolve and see whether this system is capable of making winner clones. So the first question was, can a system that is seeing such diversity select for a winner? And the answer to this is very clearly yes. This is a picture here of a cecocolonic node. It has many different germinal centers here. And you can clearly see this blue one here, which is blown up here, is composed almost entirely of CFP cells. So, and if you quantify this, you get about exactly the same 10% that you would get in an immunization. So it seems they're pretty decent at selecting, even though there is this quick flow of antigen of many different antigens at the same time. And something I'm not going to show you, there's a lot of turnover here. So GCs get invaded by incoming cells and they turn over. But even so, B cells can be selected with about the same efficiency as in an immunization. And this is just a graph here showing you that the normalized dominance score, which is the score we use to quantify how single color germinal center is, can go pretty high in 3 weeks in one of these gut germinal centers. Now, is this affinity maturation? This is a bit of a difficult question to address in the gut, but we did it in the same way we would do it normally. We did the slicing of the germinal centers and isolated these more single colored GCs. This is 3 weeks after tamoxifen, so the pictures are not as clear as what you get in the example I showed you before, which was 10 days after, 15 days after tamoxifen. But you do see these classic sort of clonal bursts happening here. So this is one example. 83% of this GC was red. Most of it came from one clone. And you could clearly see that most of it came from this variant of the clone that was really triggered to expand and leave many descendants. But we also see these other very interesting pictures where you seem to see nested clonal bursts, one inside the other, until you get to this final one here that seems to be the one that is the most evolved one. So what we could do is take these sequences here and make monoclonal antibodies for them to see, well, this is an unmutated, this is a mutated, this should be better than this one. Now, how do you assay these? There are many different ways to do it, and we did several ways. The way I'm going to show you is flow cytometry of fecal bacteria. This is a very hard assay to do, and you have to do it many times in many different mice. But it is fairly easy to compare pre-mutation and post-mutation forms of antibodies. So we see different forms of binding. So for example, this S78 seems to bind to a small population of bugs in feces very highly. And if you revert it, this population goes away, probably because it's not binding as strong and it falls back into the background. If you do this in several mice, you see this kind of picture here. And this is what we call a specialist clone. S120, the one that did nested clonal bursts, seemed to be more of a generalist. It doesn't bind as well, but it binds to a bigger population. And when you revert it, it loses binding to this bigger population again, over and over. So we can tell that there is maturation towards more binding. We can't tell you that this is affinity maturation yet, because we haven't measured precisely, and we don't know which bugs it is binding to. So how do you get closer to that? We decided to simplify, instead of simplifying our antibodies, making a monoclonal mouse, we decided to simplify our flora using this oligo MM12 microbiota that was described by the Strecker lab in Germany and given to us by Andy McPherson and Catherine McCoy. And this is a simplified microbiota that covers, that many labs are using these days. It covers 12 of the most common phyla that inhabit the mouse gut. And for our purposes, the important thing is that it's stable when you colonize mice, and it passes on vertically to the next generation. We didn't want to acutely colonize mice because that, Rolf Zinkernagel has shown a long time ago, that leads to an acute response that is different from a steady state. So we wanted to have a real steady state from F1 mice. And what we found was that you can find very nice clonal bursts also in these settings. We cloned antibodies from quite a few of them. This is one of them. We cloned the unmutated ancestor and also a mutated version of it here, M218. And what's really nice about this is because you know the 12 species that went in, you can screen your antibody for which species it binds to. And occasionally we can find them. So this one here, for example, if you do a dot plot of the different species, it binds to Enterococcus faecalis. Whereas this one here, that is another one that doesn't bind any of this. So we know that it's a specific binder to Enterococcus. And if we take this mutant here and we revert all of the mutations, this is a slightly updated version of what we published in the paper done by Chris Wickman. We see that you lose all binding to Enterococcus faecalis. So we are getting a little bit closer to claiming that this is affinity maturation because now we know that it goes from binding a bug to not binding it. And what we're trying to do now is discover what antigen it is so we can do the real biochemistry that we need to claim true affinity maturation in this system. Now, just to end, I'm going to talk quickly about one phenomenon that we found that was really surprising to us in this whole story, and we don't really have a good explanation for it, but it's, I think, cool anyway. When we were making oligo-MM12 mice, we had to go through a germ-free intermediate where we rederived brain-borne into germ-free conditions. And one thing that struck us at the time was that germ-free mice have quite a lot of germinal centers in different places. So this is comparing frequency of germinal centers among B cells in different compartments. And especially here in the jejunal and ileal mesenteric lymph nodes, you see that there's quite a lot of germinal center activity going on. We were sort of struck by this. We later realized we shouldn't be because this has been known for a long time. We found, for example, this reference from 1967 saying that germinal centers are present in germ-free animals. But the question, of course, is what are they doing and what do they look like? Now, the real surprise came when we said, let's just do a brain-borne experiment in these germinal centers. And when we looked, and this is 3 weeks after brain-borne, what we found was that these germinal centers select at a really, really extreme rate. So this is the kind of thing you can see in these. You see like really completely single-colored mesenterics and even Peyer's patches. This is very hard to see. You almost, or I'd say you never see a Peyer's patch like this in an SPF mouse. But here you're seeing really extreme levels of selection of winner clones in Peyer's patches. And if you quantify this over time, this is your 10% highly selected clones that you see in an SPF mouse. And here in a germ-free, it's more like 50% or even more than 50% of the GCs select very strongly. So what we take from this is that selection is tunable. It can be accelerated, it can be decelerated, depending on how you look at it. And germ-free germinal centers seem to be an extreme case where that shows you the extent to which germinal center selection can happen in some environments, specifically the germ-free gut. Now, what's up with these germinal centers? We don't really know. What we can see is that they are doing really classic clonal bursts. This is probably the strongest burst we've ever seen. The descendants of this red cell here that took over this entire Peyer's patch. When we made monoclonal antibodies from these, they don't seem to bind anything. They don't bind food, they don't bind commensals, they don't bind autoantigens, they are not polyreactive. And we even threw them together with PJ Utz at Stanford at an autoantigen array, and we couldn't see a single thing turn up in this clone. So they just don't seem to be amenable to finding the binding through these experiments. But there is one thing that we can say about these, that they are heavily selected based on their B cell receptor. And the reason we know this is that every mouse seems to have a version of this clone winning in their germinal centers. And this is not only the V region, but we can find these stereotypical rearrangements that we see coming over and over in germ-free mice. And this is one of them. This uses this fairly rare V147 with this ARGSNI motif in the beginning of the CDR3. There is this more strict motif, AREG-Fe, together with a V112. Both of these are pretty rare in the naive repertoire. But if you look at them in winner clones, especially in germinal centers of germ-free mice, you start to see that this ARGSNI clone shows up as red here all over the place in many mice in germ-free and also in MM12, as does this V112 AREG-Fe clone. So selection seems to be very stringent also in this, from this sense, that these mice are selecting over and over these same clones, even though we can't tell what these clones bind to. And if you need more convincing that these are very important clones, you could read Fred Alt's paper that actually described these exact same sequences before we did in a broader effort to sequence Peyer's patch germinal centers. And these are very, very much exactly the same clones that he found up in Boston. So it's not something that happens exclusively in our mouse facility. So summarizing this, gut-associated germinal centers are able to select winner clones in spite of this very high antigenic diversity. Antigen-driven maturation can happen in steady-state antigens. We are coming towards, getting towards being able to label this as real affinity maturation. This seems to be at least partly due to specific targeting of commensals in the gut. So at least in the MM12, we're pretty sure about this, and we think it's also the case in SPF. And this is sort of the interesting part about the last bit, that selection seems to be tunable. We were seeing always 10% in every model that we looked into. This tells us 10% is not something that arises because of intrinsic germinal center noise. If germinal centers are done and made in a certain way, they can select much more efficiently. And we need to understand what is it that tunes germinal centers to do this. And finally, this is a curious thing, the strong selection of these convergent public clonotypes, but we don't really understand at this point what these clonotypes are about. But what we can say from them is that they strengthen our case for very strong selection happening in these germ-free germinal centers. And instead of finishing with a model that explains to us what is going on, I'm going to, because we don't know what's going on exactly with these clones, I'm going to just end with something that interests me, what kinds of questions I think are interesting here. We know that normal germinal centers that we usually think about could be thought of as a negative feedback loop. You have infection. Infection triggers a germinal center that secretes antigen-specific immunoglobulin, mostly IgG. This feeds back into the infection and depletes the pathogen, so that we are left with, we cure the infection. What happens in the gut seems to be much more interesting. There is not a monomorphic antigen. There are many different antigens. What we seem to be able to show, and other labs are also showing this, is that there can be affinity maturation towards commensals at steady state. We're very interested in this right now, whether these strong selection events translate into antigen-specific IgA in the lumen. But this seems to be the really important and interesting question, and many labs have been working on this, is what does antigen-specific IgA do in terms of controlling the abundance of species in the gut? And this is sort of where we think we would like to go in our long-term future. So just to end, I want to thank the people who did this. I showed you a lot of the people from our lab in the beginning. I highlight Carla and Luca and Arjen, and also Tiago, who's our bioinformatician, who does a lot of the work on sequence analysis for us. We've got mice from many people, Father Inez, Jean-Claude, Tomo Kurosaki, and Tako Okada in Japan. We have wonderful collaborators at the Hutch Institute that help us interpret our data, and especially made a software for us that allows us to analyze clonal trees that we showed you repeatedly. And of course, for the last project, this was a very close collaboration with Daniel's lab, Greg, Angelina, and especially Daniel helped us, or actually didn't help us. This was really a 50-50 type of collaboration. And I'm very happy to be able to do these things in conjunction with Daniel. And these are our funders, and I am happy to take any questions. Thank you for listening.
Thank you, Gabriel. Yeah, go ahead, Enrique, please.
Well, thank you so much, Gabriel. What a treat. I mean, thank you for sharing this incredible and beautiful data on B-cell biology, ecology, and at the end of the day, immunophysiology, I would say, how this works in this setup. So I give the stage to you, Carla.
Thank you. Yes, that was wonderful, Gabriel. Thanks for such an elegant approach to fundamental biology. It was a real treat. So this is very brief, just to remind everybody that you can ask questions, but we do this through Twitter. If you go to the Global IMMUNOTALK Twitter account, you will find a tweet that says, ask questions for Dr. Gabriel Victora here. And please remember, when you ask the questions, you can reply to this tweet and mention the hashtag Global Immuno and at Victora Lab, which is the account that Gabriel is going to use to answer his questions. So thanks everybody for joining again in a wonderful Global IMMUNOTALK, and we look forward to seeing you next week. Thank you. Bye, Gabriel. Bye, Enrique.
Bye, thank you. Thank you very much.
Bye-bye. Thank you so much.